So really pleased to have this opportunity to take part in this symposium. And what I've discovered is that almost everything I'm going to tell you has been anticipated by one or another of the speakers. And in fact, I can even tell you after talking to a few of you that prospectively some of you will be talking about similar topics. So cognition, like consciousness, is one of these slippery things that seems to change with time. We all have an intuitive feeling that it has something to do with memory and has something to do with attention, and that it somehow it's the integration of information with the past and making predictions. I'd like to start with an observation, and this is a book chapter that Pat Churchland and Rama and I published back in 1994, and it was under the title, A Critique of Pure Vision. It was Pat's title. And the question was whether the common sense notion of vision that we all carry around with us, and in fact is being instantiated by engineers developing computer vision systems, whether or not that really would apply to the human brain or not, or animal brains in general. And the idea was that if you go and look at what they're trying to do is to develop an internal model of the outside world based on images or movies. It seems eminently reasonable, but if you actually go and look at the evidence, it turns out that it falls far short of what the actual evidence teaches us, both in terms of psychophysics, physiology, anatomy, and we compile a long list of things, and at the end we finally concluded that, in fact, vision cannot be isolated from the rest of the brain. It's really part and parcel, and that really the purpose of vision is to help the motor system, ultimately. But I want to give you one demonstration that we cited in our paper. This is change blindness, and some of you have already seen this. But what I'm going to do is to show you, this is an image I hope you all can recognize. There's an airplane, there's soldiers and so forth. You know, it's easy to parse the picture. What I'm going to do is to flash the picture on and off, and something is going to disappear and reappear. And I want you to put your hand up when you see it for the first time. Now, this is an intelligence test. I'm going to be looking and seeing who are the people who put their hands up first. And if you've already seen this, don't put your hand up because that's biasing the statistics. Okay, so here we go. Bump, 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 bump 2, bump, 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 bump 4, 5, 6. How could you have missed that? I mean, the majority of you really didn't see it until it was pointed out. And what's interesting is that after it's pointed out, you can't not see it. So what this illustrates is that we don't have, at any given time, a complete picture of the entire scene. And in fact, we have a relative limited bandwidth in terms of what we can represent, what it is we're paying attention to. That's represented and fully processed, but the rest of it is some kind of sketch. And unless you're actually paying attention to it, you're not going to see these changes. So that immediately tells us their intuition may be not a reliable source of information about how either our visual system or the rest of our brain is going to work. Now, most of what we know about the brain in terms of the visual system comes from recordings from single neurons. And in a classic paper by Hubel and Wiesel, 1962, they reported for the first time the existence of neurons in primary visual cortex of cats, which had receptive fields, regions which if you stimulated with light, you'd produce a very strong response that was elongated rather than circular, as you find in the retina and in the thalamus. So this suggested that there are something that looks like it's detecting edges or the boundaries and so forth. And this was the experiment that launched 1,000 microelectrodes into the cortex. And where are we today? So I'm going to summarize 60 years of research, recording from 24 cortical areas. And this is an example of an experiment in which the animal, this is a traditional experiment, the animal is sitting there, it's going to have to discriminate an object, it's going to tell you whether or not it's a familiar object. and it's got to do it as quickly as possible. And this animal's been overtrained at the task, right? So the animal's doing it really fast, really well, probably faster than you could. So the object appears, and within a few tens of milliseconds, the information is encoded in spike trains, which then get transferred to the primary visual cortex where it gets analyzed. We know the simple cells, the complex cells, topographically organized. And from V1, there's a whole sequence of visual hierarchies going to areas V4. And finally, it gets to the inferotemporal cortex. And that takes about 100 milliseconds, which is a substantial fraction of the whole reaction time, right? So half the time is spent just analyzing what's there. And from the inferotemporal cortex, that information then goes to the prefrontal cortex where we think decisions are being made. And from there, motor planning. Motor cortex has to decide what hand is going to be used. And ultimately, the acetylcholine is actually released from the muscles at the end plate regions. So this is clearly a very, very simple idea of a feed-forward pathway, how the information gets processed and decisions are made. What's wrong with this picture? Well, in fact, it turns out that this is, not only is it a wrong picture, but it basically leaves out the most important parts. And so it was already mentioned today that even before the stimulus appears, the brain is spontaneously active. The animal is expecting the stimulus. So there's a lot of activity that's going on continually through the cortex. So the stimulus appears, and indeed there is a wave of activity that does go through the cortex, but immediately, in fact, at the same time it's going up, it's going backwards, because every single feed-forward projection has a feedback connection. And that means that there is information that is flowing in both directions. So we know that the feedback projections are particularly important for attention. And the spontaneous activity is going on all the time. This is a little bit misleading because, in fact, we know that there are these default regions and resting level activity. These areas are actually talking to each other, so the spontaneous activity is going up and down at the same time. They're correlated. Okay, so the message is that we have two fundamental streams of control here. We have the visual information, which is clearly going into the system, but we also have information control that's coming from the very top areas of the brain, the prefrontal cortex, and is regulating the flow of information, right? So there's, people sometimes loosely call it the bottom up and top down. I think that's a little too crude, but nonetheless, let's use that as kind of a metaphor. So here is a classic picture, and I think that without any trouble, I think everybody in here can distinguish whether or not this is male or female. If you think it's a male, put up your hand. No takers, okay. It could have been a transvestite child, right? No? Okay, so can anybody tell me what part of the world this child is from? What is that? Russia? Did somebody say Russia? Yes. Oh, okay, you're right. So this is from Yarbus, actually, a very famous study that was done by Yarbus over 50 years ago. And the question is, when you look at this picture, you can tell roughly what the age is. What age would you think this is? 7, 9, so young. Okay, but the point is you can learn a lot of, you could deduce a lot of information by looking at this image. And the question is, what are you actually looking at? And so here's what Yarbus discovered. He put a little mirror on the eyeball and was able to track the gaze. So we make these saccadic eye movements, which are very fast, point-to-point movements, about 3 times a second. And so here is the trajectory of someone who is looking at this face, and it's not random and it's not uniform. It is very highly focused here around the eyes and the mouth area, a little bit at the top. It's clearly you're sampling information. You're seeking information from this image. And not only that, but if you take a complex scene like this, famous picture called Unexpected Visitor, there's a scene, something's happening. If you ask someone to tell us what the ages of the people are in the room, they're not sampling it randomly. They focus their gaze on the faces. There's a lot of information about age in faces. And every single question you could ask gives rise to a different gaze pattern. So if you want to estimate the circumstances of the family, you look at the tablecloth and the pictures. If you want to know something about the clothes that the people are wearing, right, you look down at the the whole body. So there's something going on here in which the goal or the task is having a profound influence on modulating what the motor system is doing in order to cue the information. Where does that come from? How do you know where to look? And are you actually even thinking about this? We're not really aware that we're moving our eye around 3 times a second, but nonetheless, it's happening all the time, sampling information from the world. Now, one of the beauties of the oculomotor system is that we actually know most of the important centers for both the visual processing and for the motor system. And I've summarized them here for a monkey brain, but it's very similar in your brain. The key structure which helps direct the motor system is the superior colliculus. It has a topographic map, tells you where you should move your eye in the orbit. This gets input directly from the eye, but it turns out that the more important input is from the primary visual cortex, in fact, from several visual areas. The parietal cortex projects down, and then a huge projection down from the prefrontal eye fields, frontal eye fields from the prefrontal cortex. And roughly speaking, the V1 inputs, early stages of inputs, are there when you're directing your eye to a bright flash. So this has got a reflexive movement. But if you're making a voluntary movement, in which you're actually asking questions, top-down questions, then prefrontal inputs are the ones that are dominating. So we have these multiple systems we have that are integrated and are working together all the time. Question is, how do you separate them? If we want to understand something about cognition, presumably we want to understand something about the top-down system, right? But it's always being mixed together with signals coming in from the outside, and so you're always you have a problem of separation of how is it that you're going to mix, what contribution is each making to any particular decision. What we need is a task in which we've separated those 2 different streams. And I'm going to tell you now about one of these tasks. So this is a traditional psychophysics task. We have a bite bar. You have a subject that is looking at a screen. There's an eye tracker, infrared eye tracker, that can determine where you're looking. A lot better technology than the Arbus had available to him. Pretty accurate, less than a degree. And here you have the screen. So here's the task. Devilishly simple. Fixate for 300 milliseconds, 20 by 20 screen, and then the screen goes blank. That's it. That's the stimulus, right? The task is to look for a hidden target. Somewhere on the screen is a location which, if you look at it, if you're within 1 degree of it, you'll get a reward. Okay, so you start looking around randomly, and eventually, by chance, you happen to hit it, right? You have 20 seconds, and then the question is, you get a new location. So you have to keep, every time you start over again, it's a different location. Okay, so what do people do? Now, the beauty here is that any pattern you see, any learning that takes place has got to reflect internal top-down, right? There's no visual stimulus that's guiding you. So here's the behavior. So at the very beginning of the trials, these are fixation points during the first few trials. You can see they're distributed all over the screen. You don't know where it is. But by the 4th trial already, and certainly by the end of the trials, you can see that the fixations, the very first fixation goes to the upper left corner. And the reason is that we were picking the locations not randomly, but rather from a Gaussian distribution that had a center in the upper left-hand corner and had a standard deviation of 2 or 3 degrees. So in fact, the subject within 1 session had learned to make eye movements that were roughly matching to the distribution of the hidden targets that they didn't know about. In fact, if you ask them consciously, they often don't really, they may tell you vaguely it's somewhere in the upper left-hand corner, but they can't tell you what they were looking for. Now, we manipulate, and by the way, I should say that this is work that was done in collaboration with Leon Tchaikovsky, and I'm going to be telling you some about the work with Joe Snyder and Mike Moser a little bit later in terms of modeling these results. But it's really intriguing because here is a learning task that can be demonstrated. Very, very rapid learning, and in fact, it's very sensitive to the parameters. So what we did was vary the standard deviation of the target distribution here, and we looked at the standard deviation of the horizontal eye movements. So the question is, is there a relationship? And so here's 3 individuals, and you can see that there's a roughly linear relationship. They have different slopes, but the fact is that people adapt not just to the location, but to the width. So they hunt and peck around, not just at the same place. Here's the speed of learning. So the first few times, in fact, you may not get it within 20 seconds. And so the number of fixations that you need drops rapidly. In fact, after the first 10 trials or so, you've reached an asymptote. And I'm going to come back to this a little bit later. So the blue here is the human performance. And this is this is over roughly one session. Not only that, you learned a distribution. So here's the distribution of standard deviation of the saccades, and you can see that it's very broad at first, and then within the first 10 comes down to about 2 to 3 degrees. Roughly the width of the target distribution. Okay, well, let's try to understand what's going on, right? Somewhere in the brain there must be some place that's keeping track of the previous experience you've had, and it's using that to guide your eye movements in the future. Well, we have actually a very nice model that accounts for all of the data that we have. It originates from Andy Bartow and Rich Sutton. It's a method called temporal difference learning, and I'm not going to go into the details except to say that there's a perception-action cycle here. The environment puts up some stimulus, which causes the cortex and the rest of the brain to achieve a certain state. And on the basis of that information, the brain has to make decisions. And this is a reinforcement learning model. The first thing, you have to make a decision about what action to take, but you're making that on the basis of a value function. You have to evaluate the particular stimulus and what's important. And on the basis of that, you take an action, and on the basis of your action, you may get a reward, and on the basis of the reward, you update your current value function. So you update it so that the next time the stimulus comes, you're more likely to go for it if you got rewarded, or if you weren't rewarded, you won't go there again. So that's the general viewpoint. Where is this in the brain? Well, a lot of evidence that this actor-critic model with the value function and the policy are in the basal ganglia. That is to say, the basal ganglia computes the value and the associated action that should be taken. And the temporal difference error here has been associated with a neuromodulator, the dopamine neurons, in the ventral tegmental area. So here's the dopamine system, and we think they're computing the reward difference, that is to say, the prediction error, and reporting that to the basal ganglia and to the entire cortical mantle. Very broadly distributed. Now, how do we know it's a temporal difference? So the temporal difference has a signature, which is that you have to subtract the expected value from the actual reward, and it's the difference that you're using for the learning process. And if the dopamine neurons are actually doing that, then we should be able to capture that. So here's an experiment that Wolfram Schultz developed where the monkey has a simple task of reaching for a reward, and there's a light that goes on that predicts whether or not the reward is there. So there's a light that goes on. At the beginning, there's no light, and the reward, you can see the neuron responds weakly to the reward. After pairing the conditioned stimulus, the yellow light, with the reward, two things happen. First, the conditioned stimulus itself produces a response, and the response at the reward disappears. Now, according to temporal differences, this is actually, this lack of response isn't because there's not a signal there, it's that two signals have canceled. So on catch trials, what you do is to, after the animal has learned the task, what you do is to present the stimulus but withhold the reward. So the reward doesn't come, and it should have canceled the prediction, but it didn't, and so you can see there's an inhibition here. There's a lack of spikes because of the fact that there's a failure of prediction. Let's apply this actor-critic model to our model. Very simple. We have exactly the same setup in terms of the reinforcer. We have the value function. We had to add one additional factor, which is the fact that humans and monkeys tend to favor short saccades. So you typically look around in a neighborhood and then make a long saccade every once in a while. That's a pretty good way to search, by the way, because you want to go to a completely different place to start looking and hunt and pecking around a particular location. So if you put a penalty in for distance, you can very, very accurately reproduce the learning curve. So here is the reinforcement model, the temporal difference model in green. And you can see that it tracks the human really accurately. But there's one additional analysis that we can do, which is, what can the ideal observer do? If you took all the information that's coming in and you had to do the best you could possibly do using a Bayesian criterion, how well can you do? And that turns out to be this dashed line here. And this is what Joe Snyder worked out. So it turns out both the humans and the model achieve, within a very few number of trials, the best you could possibly do. And it's not just achieving the best performance in terms of number of saccades, but also it also matches the optimal search strategy for how far you should be hunting. So this is, which turns out not to be the standard deviation of the Gaussian. So that's interesting. It's a little bit bigger. Okay, well, how is that dopamine signal actually getting into your decision process? Somewhere along the line, that information has to be taken into account to guide your behavior. So this is an experiment that was done by Paul Glimcher over 10 years ago. And what he showed was that if you have a monkey that is just making a very simple saccade to a target, a light that goes on either to the right or the left, that you can change the performance of neurons in the parietal cortex. These are visually stimulated neurons, and also neurons that have an action component. That is to say, they fire just before the saccade. So these are integrating sensory and motor. But if you change the amount of reinforcer, the amount of juice, in this case in block 1, you can see there's going to be 0.2 of a milliliter to the right. And in block 2, it's going to be half of that, and it'll be 0.2 to the left. And now just look at that single neuron that they recorded for the entire duration of both blocks of trials, and focus here first on the bottom. And this is the block in which the stimulus was 1/10 of a milliliter to the right. And you can see that there is a very vigorous response here after the stimulus comes on. This is the visual response, 25 spikes per second, and a motor response. Now, very same visual stimulus, the very same motor output, but now you've simply increased the amount of reward by a factor of 2. And now look at the firing rate of the neuron. This visual stimulus here, when it comes on, is increasing the firing rate by a factor of 3. No change to the motor side. So somehow, this neuron is reporting not just the fact there's a visual stimulus, but the fact that this has a certain value for me. It has twice as much value in the red compared to the blue. And this is actually one of the main points that was made in the critique of pure vision. How could it be that there are neurons in the visual system that are telling you about value if all they are supposed to do is recreate a model of the world? The value of that particular object is going to change depending on the contingencies. So here's the parietal cortex. So we know that the parietal cortex projects directly down the superior colliculus. So this is a possible pathway. We have trained monkeys to do exactly the same task. And it should be possible now to record from all these different areas and track the signal down. Where is it coming from? But now here I want to very briefly, how much more time do I have? Okay. Tell you a little bit more about the anatomy. And this is going to be a little bit of an overload for you who don't know basal ganglia. But the basal ganglia is a very ancient structure present in all vertebrates, and there's evidence for homologs, real homologs, in invertebrates. And it has more than one job, okay? And I'm going to be going into that in some detail. But it gets input from the entire cerebral cortex, and different parts of the cerebral cortex project to different parts of the basal ganglia. It projects to the striatum, as you can see here, several pathways that take you down, and then to the thalamus, and from the thalamus back up to the cortex. So there's actually a loop. It forms a loop with the cortex. And in fact, once it forms a loop, you really don't know where the information has come from or where it's going, because it's basically part of the same system, right? So I mean, people who are cerebral cortex centric don't recognize how much of the data, how much of the activity in the cortex really is a consequence of these loops. But there's also a very strong output to the superior colliculus. And so it's clear that the basal ganglia has a very important role to play. In particular, it's known that this is inhibitory, so that means that it has a role to actually suppress eye movements. Okay, but now I want to focus on this box here and look at it from the perspective of these different parts of the cortex. So if you look at the sensory motor part of the cortex, which is in the back of the brain, we can see that the sensory input from the visual system, somatosensory system, project down and have direct output on the the regions of the brain which are going to control actions. And so this part is thought to be important for sensory motor associations, sensory input giving rise to a motor output. So this is a very simple reflexive response, and clearly it's important to have them. But what's more interesting is that there are other parts of the basal ganglia that are more deliberative. That is to say, this is the part that comes from the parietal cortex, for example, that I showed you in one of the previous slides. And here, the associations are not with the actual motor output, but with the goal. So it's really the outcome that's important here, associating a particular state of the parietal cortex, sensory and motor, together, being associated with a particular goal. And it's this loop here, which is probably the one that gets elaborated in humans, that allows us to plan ahead. And it's known, again, through many rat experiments, that over time, this loop here gets downloaded into the sensory motor loop. So something that is deliberative at first, over many, many trials, becomes automatic. And for some reason, because of the fact that this is called a habit, forming a habit, the basal ganglia has gotten this undeserved reputation as being a kind of a dumb part of the brain. Far from being the case. The basal ganglia actually may be the smartest part of the brain. And I'll get to why I think that in a minute. Now there's another limb of the basal ganglia, which is a very important one for overall importance and guiding us toward survival, and that is the limbic network. This is the one that gets input from the hippocampus and the prefrontal cortex, orbital prefrontal cortex, ventral prefrontal cortex, and it really has a very important role to play in terms of guiding behavior that is either going to be important for survival, either to approach or to flee, or what to do under certain circumstances that will help guide the rest of the brain to take action. And so there's if you look at it from the perspective of gradients, habit formation tends to be, it's just, by the way, we broke this into three. There's actually probably like 6 streams, and they're overlapping. So it's not like they're separate modules. But there's a gradient. That is, say, more to the right is more habit forming, more specific for the effectors, particular muscles, and more automatic. As you go to the left here, it becomes more cognitive, depends more on deliberation and increasing effector independence. So there's a very interesting linear, maybe not linear, but at least differences in the function depending on which part of the brain and which part of the striatum are processing the information. Okay, now I want to take you back in the time machine to 1971. How many of you here remember reading a review in the New York Review of Books by Noam Chomsky? It's a very famous essay which was, okay, I knew that, which was against B.F. Skinner. And, okay, so this is an essay which had enormous impact on I think on the history of the field, especially in cognitive psychology, and ultimately, leading to the symbol processing and rule-based approaches. And the argument went something like this. B.F. Skinner has been working now for decades on reinforcement learning, conditioning, classical conditioning, instrumental conditioning, and he's gotten these rats to do some pretty amazing things, and the birds are even more impressive. But I can't imagine learning language with that weak of a learning system. We've got to take many trials and so forth. And if I can't imagine it, then neither can anyone else. So it's impossible. And language must be innate. And literally, if you boil down the argument, it's an argument from ignorance. Right? I can't imagine it happening. It can't happen. Okay? So I want to give you a counterexample. How do you prove someone wrong? You give them a counterexample showing that actually you can get it to do something really sophisticated that you couldn't imagine because you weren't smart enough. Okay? So this is a game. Jerry Tesaro worked in my lab as a postdoc back in the 90s. This is work he actually did when he left the lab and took it to IBM. And his thought was as follows, that temporal differences is an algorithm, it's a reinforcement algorithm, but it's not just for associating reward with sensory input. Actually, it's an algorithm for solving a control problem in engineering, which has to do with how do you optimize reaching a goal when there are many steps that are involved, and you don't actually get the reward until the very end. So you have a problem. You get the reward, you get there. How do you know which of the steps was crucial? That's called the temporal credit assignment problem. The temporal differences learning algorithm actually solves the temporal credit assignment problem. And obviously, this is going to be important for animal behavior because animals don't just get the reward immediately on a lever press, except in the lab. Normally, they have to go through the world and make decisions to get there to the right place and do the right thing. So here's a simple game now where we can actually apply it and see how far we can take it. This backgammon. It's a very common parlor game. People play this for money. There are world champions. It's a huge business, okay? And people are really good at it. So the idea is, how many people here know about backgammon? So I don't have to go through the details. Okay, at least half of you. Very briefly, the idea is in order to win the game, you have to take all of your men all the way around and then bear them off. And the other, your opponent's trying to do the same thing in the opposite direction. And what makes it a particularly difficult game to play, that's say to predict, unlike chess where it's deterministic, there's a probabilistic element. You have to throw the dice. And depending on the numbers that come up, you can move your men ahead by that many positions. And there's also complexities having to do with the fact that if you have two of your men on a particular point, then you can't get knocked off, but if only one, you can knock them off and go back to the beginning. So there's a lot of strategy involved here, specifically at the very beginning where you're setting up defenses and then you have to decide when to become aggressive, right? With that moment where you have to run for the exit. So Jerry set a program up in which the input, the cerebral cortex of his model, was the game position where they're located, and he had specific explicit representations when there are two or more. So he engineered in a really good representation for this game. But then he just turned it on and let it play itself. And at the beginning, it flailed around, completely stupidly moving forward and backwards, and eventually it ended. One of them won. Right? And then on the basis of that one little reward that you get at the very end, he changed the value function, right? Temporal difference learning. And he let it play over and over and over again. And so here are the results. After 300,000 training games, it was beating him. He was a strong intermediate player, but somehow it had discovered ways of getting around him. So he brought in some experts from New York City, in particular Bob Roberti, who has written books on positional play and backgammon. And in a total of 51 games, it lost 1 point per game. It lost on the order of a quarter of points per game, which is pretty good. In fact, what they told him was that this is probably the best backgammon program that's ever been written, right? That this was amazingly good. In fact, they actually wrote down the games and took them back and analyzed them and discovered that T.D. Gammon had come up with moves in particular positions that no human had ever thought of before, which were better than the best position, the human wisdom. Well, the beauty is you can just keep playing over and over again, and by the time it got to 2.0, it had played almost a million games. And again, by this time, the backgammon world had gotten very excited, and they heard about this program, so they wanted to come to IBM. And so they played 38 games, and now it was losing only 0.18 points per game, which is, it's getting better. And then finally, after playing almost twice as many games, Bob Roberti decided he'd come back and played 40 games, and it basically played him to the standstill, right? This is basically noise, 0.02 points per game. And at this point, he was absolutely astonished that it could reach it. And he said that at this point in history, it could easily have won the championship, world championship. And the reason is it's actually a lot of fluctuations, and sometimes there's luck involved. But he thought that this was probably as good as the best humans at the time, which is really astonishing because if you just think about this, this is just a reinforcement learner. There's no rules in here, right? There's no complicated reasoning. It's simply it had actually creatively discovered positions and moves just on the basis of the experience. And let me tell you, if you think a million games is a lot, right? Well, there are on the order of all the possible games, there's about 10 to the 20th possible games. This is only 1 in 1 millionth of 10 to the 21. That's a drop in the bucket. It's not like it memorized every possible game. It's on the basis of a few games, it's able to generalize, to play new positions it's never seen before. And that's really what human intelligence and your ability to adapt to a changing world has to endow you with. Otherwise, you're not going to survive. You can't just memorize things. So, what about evolution? Now here, we have a beautiful story, because it turns out that temporal differences was discovered very, very early on. Classical conditioning is found in almost every species. And the champion learner is the honeybee. And Peter Dayan and Reed Montague, who developed applied TD to the dopamine system when they were postdocs in my lab, also applied it to the honeybee. The honeybee is very good at being able to distinguish between smells, shapes, and colors of flowers, and it's able then to very rapidly change its preferences as the season progresses and different flowers go into bloom and out of bloom. And so it has to really be able to, almost on single trials, to change its behavior patterns. So we applied temporal differences specifically based on the anatomical organization that Randolph Menzel had discovered. He had recorded from a particular neuron in the honeybee called VMX1 and found that it, just like dopamine, the cell, it was actually responding and predicting the future rewards. And so this is a really interesting, much simpler system, but nonetheless, it had a lot of the basic elements that we saw in the mammalian basal ganglia circuit. These neurons project very broadly to the sensory structures, to the mushroom bodies, which are thought to be homologs to the hippocampus in the bee, and to the motor structures. So it's in a position to really provide very important information on and affect behavior. So just to make a long story short, I'm not going to show you the data. It had been shown earlier by Leslie Reel at the Indiana University that bees tend to be risk aversive. If you ask a bee to choose between a constant half a microliter reward and 50% 1 microliter, 50% 0, they'll always, well, with a high probability, not always, choose the constant reward. So they're risk aversive. And as you increase the variance, they will become more risk aversive. Now you can balance it by increasing the average reward of the one that's varying so that it balances. And that particular relationship is one that we nailed almost perfectly with this very, very simple model. Right? So we think that the algorithm itself, although instantiated in different ways in different species, may be carrying out the same function whether you're a bee or even a human. And so this may be something important in nature, is that you start out with a simple algorithm that is successful, it works, so you don't throw it away and start over again. You don't want to put in a super algorithm. Stay with the algorithm, but what you can do, and this is what's happened in primates and especially in humans, you give it a much better state representation. In other words, the cortical representation, which helps make decisions and guide behavior, is much richer in the human than it is in the bee. We can make decisions not just on the basis of the smell and the color and the shape, but on very subtle changes and differences and social interactions and so forth. So it's clear that by adding in a much better memory system, a much better way of representing with working memory and being able to prospectively imagine future, all of that can serve as much a better input for a value system that is going to evaluate how good would that action be if I went out tomorrow for dinner, right? Would that be something that would be better than having lunch, going to a movie, right? So you have to make these decisions every day on the basis of your imagined reward into the future. Okay, so I think that takes us to the very end. In fact, this makes the point anatomically. This is the ventral medial prefrontal cortex. This is the part at the top of the basal ganglia hierarchy. That's the one that's making the most important decisions in your life, not just whether to go to dinner, but whether or not to marry somebody or to go to graduate school or take a job. Right? These are very important decisions. Well, it turns out that these areas way up in the prefrontal cortex here that are colored, in particular this one here, at the very tip, area 10, has very strong connections with not just the other areas of the prefrontal cortex and the cingulate areas, but also the hippocampal formation. It's getting information about specific events and episodes in the past. It's getting information from the hypothalamus and telling you something about the importance of food and safety. Has projections to the the cortical thalamical loop, this that we've been talking about, focusing here, and all of the modulators, neuromodulators that affect attention, behavior, rewards, and discounting. All those neuromodulators very richly invest the prefrontal cortex. In fact, there's a gradient in which there's a much stronger dopamine projection to the prefrontal cortex than to the back of the brain. So if we put this all together into a picture, I think what we can see is that we have the same ground plan. We're not going to throw away the basic mechanisms that have been important to get us here, but we've supercharged it with an incredibly powerful representation, not just of the sensory state, but the past and projected future. And that is what is allowing us, perhaps, to continue to develop, through culture, more and more sophisticated conceptual frameworks, and with language, ways of passing it on to the next generation. And ultimately, and I usually start with this slide because Pat Churchland and I, when he wrote The Computational Brain, decided this was a good way to organize the different levels of organization in the brain. If you really want to look at the problem, and as John Searle put it, understand the basis of consciousness, we're going to have to understand everything, all the way down to the molecule level of where are those molecules located, which synapses, which part of the basal ganglia, what's dopamine doing. This is really the basic functional architecture. And if you're looking for differences with humans, I would look up here at the higher levels. We know that the molecules, these ion channels, go back to bacteria. I mean, they've been fiddled with in the sense they've been duplicated and specialized, but they basically were discovered a long, long time ago. A lot of the networks, principles here, very common, all the way down to invertebrates. But really, when you come down to the highest levels here, it's really the place where we see the most evolution taking place. So let me just end by thanking my collaborators, both present and past, and hopefully the future ones will be on soon. Thank you.